So what I'm going to do is just walk a little bit through this module that we designed to go through the principles of DNA sequencing. And then there's a student activity that's supposed to be in the module, but because we are still programming the activity, I'm going to do this with a PowerPoint slide. And in fact, I'll put the PowerPoint slide also onto the shared drive. So just in case, you can always use the PowerPoint to do this activity. So here in the first slide, first page for DNA sequencing, we just simply go through the rationale for DNA sequencing, the idea that the sequence, that the actual sequence of the molecules gives us the information, that, and we want to know this information. The next button then goes to the technical challenges of sequencing. And as I mentioned before, one of the key concepts I think is to get through to students is the scale in which we're trying to measure things. Right? So as I already said, the example of our hand being the size of the continent of US, if we're trying to look at individual DNA molecules that are the size of a grain of rice. So the challenge is really to try to come up with ways to measure things that are at this such small, tiny molecular scale. This building here is the nanotechnology building. And as you know, the word nanotechnology is something that came into vogue in the last 10 years or so, and students are also interested in this idea of nanotechnology. Biologists have been doing nanotechnology for about 40 years or so, because everything we try to manipulate for the organism really is at this nanoscale.
Is this called a synthesizer?
This machine here,
yeah,
is called a sequencer.
Sequencer.
Yes.
So it makes the sequence, and how do you analyze that your sequence is correct?
How do you analyze the sequences? You mean how do we know after when we do the sequencing reaction that the sequences are correct?
Yes.
So we do that. We know if the sequences are correct up to a certain amount. So we can't guarantee that the sequences are 100% correct. Okay? And the reason for that is that these are chemical reactions, right? As you know, you can't guarantee your chemical reactions to be 100% accurate. But we do know, for the most part, that the sequences are, say, 99.99% correct. And in fact, the sequencing machines will actually report to you not only the information on the letters, but what it thinks is the correctness of that letter. Okay? So it will actually give you a quality for the letter. And we know those qualities by doing lots of other control experiments. For example, sequencing the same piece of DNA, not only using this machine, but different kinds of techniques to sequence it. And using that, we can cross-validate that this machine will, in fact, repeat that experiment with, say, 99.99% accuracy each and every time we run it.
Do you use mass spectrometry?
You can use mass spectrometry to do DNA sequencing. That would be much more involved.
Yeah.
There are ways to do it just purely chemically. Okay? And there are ways to do it using the same kind of chemistry that the machines use, but except just one DNA piece at a time. These machines, as I said, do billions of DNAs at the same time, which kind of, you say, okay, it's doing it 1 billion times, maybe it's going to have more problem. But we can isolate the chemical process and do it one piece at a time.
Just out of curiosity, what does a unit like that cost?
$500,000.
I'm going to guess $1 million.
So a few $100,000. So there are cheaper ones that are somewhere around $200,000 to $500,000 or so. To be honest, I have no idea what the real cost of the machines are. Anytime somebody comes out with a new technology, they seem to start at this number 100,000 and up. So, all right. So there's a little bit of history of sequencing, but of course, if you want to expand on it, there's a huge amount of information on history of sequencing. Actually, Wikipedia is always pretty reasonably accurate source for many of these kind of topics. So the main point here is that the machines that we use today up here run the same kind of chemical reactions that was originally invented by Fred Sanger, the Sanger sequencing by synthesis reaction, except we do it billions of reactions at the same time instead of just one by one. And so these things have now just called next generation sequencing machines. The reason is that first came the manual sequencing that I used to do when I was in college, and then there came automatic sequencing machines, which took a lot of the manual operations out of the way, but it still wasn't like billions of reactions at the same time. Then about, I guess, 10 years ago, people started inventing these machines that used the microfabrication technology to be able to do chemistry, billions of reactions at the same time, and we started calling these next generation sequencing machines. Okay, so the next set of units then go over the chemical principles of sequencing by synthesis, the idea, of course, of the complementary base pairing, and then how, when you have a particular string, then there's an exact complementary sequence, and we talk about the direction of the string, so that there is, in fact, going one direction, left to right, there's a particular polarity to the string, so we call 5' to the 3'. The complementary sequence goes the other way. And then how DNA replication can happen by synthesizing or matching nucleotide to each. And so there's a little animation there that shows how the complementary sequence can be synthesized. There's a little discussion on how you first start the synthesis with a sequence called primer. And actually Scott raised the point to me yesterday, a very important point, that when in the textbook, and when you learn about DNA replication in the textbook, in the organism, in vivo, this primer is an RNA molecule. Okay? But in the technology, when we do the reactions, chemical reactions, we usually use DNA primers. Especially because RNA is an unstable molecule that is hard to deal with in the lab. So we use DNA primers. So we'll have to fix this and make a note of it, because I'm sure that the students learn the term primer in the textbook and they learn it as an RNA molecule. And so we talk about the DNA synthesis step, and you have this. And then the question, then the question is, what use is this? Because we made the point that DNA molecules are very tiny. We're at the level of a little bit bigger than the hydrogen atoms, so you can't see it anyway. How does it help to synthesize the DNA? Right? So how does it help to make the copy of the DNA? And the main thing is, when you make the copy of the DNA, you can actually modify the base pairs to put in there on the base pair things that you can detect more easily without having to look at it with a microscope. Right? So these are things that, say, emit light, or things that emit radiation. When I first learned to sequence in the 80s, we attached radiation molecules. So typically, the phosphorus molecule atom was a radioactive one, and therefore, after you ran the chemical reactions, you were able to know what base pair was newly synthesized by putting those the chemical reaction on an X-ray film, and then the X-ray film would become dark because of the radiation given off by the molecule. Nowadays, we don't use radiation anymore. We use fluorescent molecules. Fluorescent molecules emit light. When they're stimulated with a particular spectrum frequency of light, then they change color and emit light at a different spectrum, so we can detect it. And so this is like reading a book, say, at 1,000 yards, and with a binoculars, you obviously can't read it. Okay? But each individual letters, if they were to glow, and let's say that they were to have emit light themselves, but then you'll be able to detect the idea that there is, in fact, some light coming out from that book, right? And so you don't have to actually read the individual shape, you just to be able to see the light. And that's the main detection technique that we use now. Different color light that are labeled for each of the different nucleotides that we're newly synthesizing. So, the main thing that these new next generation sequencing machines do is to take these strings and synthesize these new molecules one step at a time. Right? So if this is the string that you're trying to sequence, and let's pretend you don't know it, you start your chemical reactions, and then the chemical reactions will say incorporate a G base pair complementary to the C base pair here, and that new G base pair will be labeled with a fluorescent light, and we'll be able to see it. We irradiate it with the chemical reaction with a laser, and then it glows blue, and we're able to see it. Okay? And then we take a picture.
Is that color, is it tagged on to the G? Is that how it does it?
That's right. The color is tagged on by adding a fluorescent moiety to the G molecule itself. That's correct. Now, another chemical reaction is done to take off this color from the G molecule. Okay? So the light is turned off. Okay? And then the next step, then the next base pair is incorporated. Right? Again, complementary to what's there. And it now has a different color light. And we see that now it's glowing green. And so we're able to see this. Okay? So we take a picture, we turn that off, and then we go on to the next one. And we continue to do this one by one. Okay? Now, if we were to do this by hand, this would be a very tedious chemical reaction to do. You post some chemical into your tube, let it glow, then you put another chemical, turn off the light, put another one, and you have to do this over and over again in order to get this whole reaction to go. But the machines do this by a set of technologies called microfluidics. That means that all these chemical reactions now are done as if they were complicated plumbing system in which the chemistry is done by flooding it with, say, the reaction chamber with one kind of chemical, washing it off, and then letting the next set of chemistry come in. And I'm sure you're very familiar with that kind of process in, say, drug manufacturing.
I did Merrifield synthesis, so I know that's the
Absolutely.
grandfather.
Yeah.
So.
Or say, actually, if you go to like oil refinery plants, there's like city blocks of pipes in which all these chemical reactions are happening step by step sequentially, right? Now we do this in reaction chambers that look like this. I'll hand this out to you. These things are called flow cells. So this streak that you see here is kind of a pipe tube. All the DNA molecules are actually spotted on this little strip, and the chemicals go up and through this, and reactions happen, things get washed off, reactions happen, things get washed off, and so on.
So would you do it like after it's all done, just flood it with like all the yellows, and then all the yellows will light up?
That's right.
And then you do it 4 times.
Exactly. And we'll do that exercise as an activity exercise in a moment. If anybody wants, I can supply the things that you can take to your classroom. We run through these all the time. The only thing is that it's not very impressive unless you can look at it under, say, a dissecting microscope. So if you have a dissecting microscope, if you look at these, you can see these tiny micro-engineered features that are in these glass slides. So the next concept that we try to discuss is that the light coming off of the strings that you are newly manufacturing really is not enough. You can't really see the light coming off a single molecule. So you actually have to have many copies of the molecule. So the reactions are happening exactly as we described, but happening over many copies of the molecule at the same time so that we can actually see the light. Because the light is too faint when it comes off a single molecule. So we actually make many copies of the DNA that we're trying to sequence and have those light come out all at the same time. Okay? And when that happens, sometimes you can have errors happen. So this should be all, say, G light, the blue light, or sometimes a little bit of green light can creep in. And this is where the sequencing errors can happen. As you mentioned, what's the accuracy of these machines? Well, this is a chemical process, so you can't guarantee 100% accuracy. And the important part of this is that these kind of errors creep up and accumulate as you're doing the sequencing over time. So this effectively limits the amount, the length of the DNA that we can sequence at any given point. Okay. The current technology, we can do about 150 base pairs to 200 base pairs before the error accumulates too much that we can't really detect the signal. Yes?
Now, after you've done your synthesis, do you purify them by, let's say, phoresis, or how?
No, there is no. These are actually pure DNA molecules that's already put onto those glass slides that I just ended up.
Right, but what I'm getting at is that when you're doing this, like you said, there are errors that are involved. Right. And would you say the synthesis is what, over 90%?
No, 99.99% accurate.
Oh, so then you wouldn't have to have the purification.
No, there's no purification, that's right. So it's typically about 10, more than 10 to the minus 4 accuracy.
And how much are you making in the process here?
Well, so as I said, that you do it one step at a time.
Well, I'm just saying, when you're in this well here, how much do you actually make?
So each of those spots probably contains about 4,000 molecules of the same copy.
Got it.
So this is what the actual reactions look like. So this is a picture of one of those tiny part of those that glass slide flow cell that I just handed up, and these are sort of the colors that you see. Okay. Now this is actually a synthesized image, just to be technically correct. Okay. The colors happen one at a time, okay, so you don't actually simultaneously see all 4 colors at the same time. So this is just synthesizing what would happen if you saw all 4 colors at the same time. You only see one at any given time. So each one of these dots contains about 4,000 molecules of the DNA that you're trying to sequence. The colors are different because obviously every spot has a different string that you're trying to sequence. And we're trying to sequence billions of such strings at the same time.
So maybe the answer is it doesn't matter, but would you take a genome and break it into 150 to 200 nucleotide lengths? And then somehow like this one comes first, and then that one, and then that one?
That's absolutely correct.
Wait.
And so that's sort of the concept that we try to cover next.
Okay.
Okay. So here's a string. Because of technical limitations, we have to break it up. Right? So we have to break it up and we're only able to read small pieces at a time. And somehow we have to be able to put this back together again. Now, if you were to take those strings and say, look at all those strings and put these, say, end to end, then you end up with this string, which is not the same thing as what you originally started out with. So obviously, you shuffled around all these little breaks, right? So the key problem is trying to put this all together. And we do that by taking the original genome string and breaking it in different places. Okay? So we don't just break it right here. We may say have another string where we broke it over here. Okay? So from this one, we might get this set of sequences. From the second one, we you'll get this set of sequences. Right? And then we have information that this, say, GATC comes before GATTC, GAT, because from the second cut, we have this string which bridges between the two groups, right? So that here, GAT, TCG, GATCC, GAT. So this little, the second string is like glue that tells us these two strings belong together.
So I guess it's almost like having two copies of a jigsaw puzzle, but you cut the pieces wrong on the second one.
Exactly
so you
right.
piece it together faster.
That's exactly correct. Yep. And that's a very good metaphor. So, of course, when you really get to the human genome, 3 billion base pairs and 150 base pairs each, so you're going to end up with something like 5 million pieces, 50 million pieces. Okay? So you can't do this by just looking at it and doing it by hand. So all of this happens with a computer. And unfortunately, it's not easy to do because it turns out that most animals, and especially the human, the genome, the string is not unique. There's lots of repeated pieces. Okay? And so this is like having jigsaw puzzle, as you mentioned, where the shape is exactly identical. Right? So if you have lots of pieces where the shape is exactly identical, no matter how you cut it, then you don't really know how those pieces fit in together. But this is right now one of our big technical challenges, how to do this very, very well. All right, so let me go over then to the exercise. Okay, so I did this as a PowerPoint. This should come up as an animated link once we have finished the Java programming on this. Okay, so the activity goals for the sequencing by synthesis is to understand this idea of sequencing by synthesizing one nucleotide at a time. To understand the idea of spatial indexing, and I'll go over this a little bit more in a moment, that is how the sequence information in one position all belong together, okay, and how that allows us to do all these parallel chemical reactions, and understand the problem of seeing signal against noise. So what we're going to see is these colors are going to be used to detect each kind of nucleotide. Okay. So we are imagining that we are looking at those flow cells that I just showed you, and we are going to look at about 5 spots on this flow cell. Okay. So these are the spatial positions. So what we mean by spatial indexing is that when you see, say, each of these positions have little strings that we're trying to sequence, okay? And we do it by synthesizing on those strings. So if I see this complementary letter A, I know that the string started with a T. And we're going to see these signals, and these signals are going to come on, and all these signals in the same location belong to the same string. Right? And so that's how I know that all this information right here is from this string, all the information from here is this string, and so on. So that's the idea of spatial indexing. Now these things are going to happen all simultaneously. So this is where the idea of parallel reactions or parallel sequencing comes. It's not really that just one spot starts giving you signal. All these spots are going to start giving you signal. And so the exercise is designed to teach students to track these information coming simultaneously from different spots. Okay. The spatial location of the color signal will not be obvious. There isn't going to be these dot, 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 circles around it. You just have to recognize, oh, there's a bright spot here, that's the signal, and there'll be certain other spots, and that's noise. Okay? So they have to recognize signal and noise.
And so they're not like, they're clearly not arranged in like a grid. They're
not arranged in a grid. In fact, the actual chemical synthesis of those little tiny little points happens randomly. Okay? So that you remember that sort of picture I showed. All the dots are at various different places. Okay. So there's the spatial grid. Okay, and what's going to happen is that we'll pretend that the flow cell gets flooded with this red chemical that stands for letter A right there, and what gets left is these spots. Okay? And so the students have to write down, and if you want, you can do it yourself, write down on a piece of paper, this spot there was an A right there. Okay? 3 spots with A's. Okay? The next round of the chemistry will happen, and then you get, say, another spot set up for the C. So this spot, there's a C. Okay? And then here's a blank, so that means that there were no G's in this area. And then you get T. Right? That was one round of figuring out the first base position for those 5 spots. Okay? Now, we are going to try to have a Java program to allow students to sort of click a button and take a picture of these, so that they'll have an accumulated stack of pictures that they can go back to if they want to. And this is, in fact, what actually happens in the sequencer. The sequencer goes and irradiates those flow cells with a laser and takes pictures. And then we repeat this process and going through these cycles, and the students should be writing down all the information they see as these cycles go through.
Are you going to have a sheet with the 5 circles on there to write the letters in?
You can have a sheet. I guess that depends on how much information you want to give the student. For example, in some ways, the idea that there's 5 distinct spots in of itself is something that should be deduced from this experiment. Okay. And so, because that's what we do. In fact, the machines actually, they don't know where the spots are. They scan it, they take the picture, image analysis program so to figure out, oh, there were 5 spots here. And here are the locations of the 5 spots. So you can choose to give them the information by actually drawing the circles as a template and handing them out on a piece of paper and asking students to sort of track it and write down the information in those 5 spots, or simply that they need to figure this out.
Well, the real flow cell, right, there are like 8 lanes, each with several thousand circles where you would put each string, right?
Yeah.
So they do have a.
I think the same that there is.
No, actually, so we know there are 8 distinct, sometimes there's 2. The ones that I gave you, was there 2 or 4, I don't think. They come in different lanes, and those are just so that you can run different samples on them. But within any given lane, there isn't a grid. The molecules are randomly deposited in several billion spots on those. And as I said, the pictures are taken, and it's only after we take the pictures we figure out where the molecules are.
Why are there spots at all? Aren't they all kind of mixed up together? How fine is like the measurement to separate the spots out?
That's a very good question. And so that actually then gets to the very details of how these slides are manufactured. So what really happens is that on these slides, a little bit of oligonucleotides are pasted all over the slide. So they are just deposited randomly in a certain amount of density along on the flow cell, and so they are all the same. So you can think of these as little hooks that are all over the spot. Then we take the DNA that we want to sequence, and they're all chopped up, right? And then we adjust their dilution, so there's a certain amount of molecules per microliter, and we flow it across this. And then these hooks randomly capture the DNA in single molecule. Okay? And we try to make the actual concentration such that it's not, if it's too high, then every Velcro hook is going to grab our DNA. And when we image it, we won't be able to tell the difference. It's like all the spots are all filled, right? If it's too dilute, then we're not running a very efficient experiment because let's say there's only 5 spots and I wanted 1 billion. That's a lot of wasted money, right? So we try to adjust the concentration such that just the right amount of density of molecules are captured. And then chemical reactions are run so that once you capture that molecule, copies of that molecule is made right on the spot. Okay, so most of you know the PCR reaction, okay, and that they are often covered in the high school technology sessions as well. PCR reactions are made to happen right on that surface. So the copies are made right there. And that's how the spot gets formed on the surface. Okay, so once we complete these cycles, then there will be, so, you know, I've done this exercise, by the way, with high school students many, many times, and the amount of cycles you want to have and the number of spots you want to have really depends on their attention span. Sometimes it works really well, and I'm sure you have the same experience as I do. 1 year, for whatever reason, students love it, works really well. Next year, it's just a complete dud when I'm doing exactly the same thing, right? And so that will happen. And so, you know, at some point, you can choose to stop. I think having strings that are few letters long, more than few letters long, does give the students the idea of the complexity of the process. And about 5 spots is okay. Okay? So then eventually they are supposed to understand that this was the starting string. And you ask them, to, okay, what are the strings? You read it off and you give them the answer. Okay, so that's all we have for the DNA sequencing.
And what about putting it together in the right order, though?
Well, so that's the bioinformatics problem. And so I hesitate in making that actually exercise. We do have some exercise in the bioinformatics session because it's a very tedious process. You know? I have done that in class by giving them physical strings. So I've printed it out onto a piece of paper and cut them up in different ways and gave it to the students as a puzzle to solve. That actually works much better because then they can use physical things to move things around and they're able to come up with the right string. If you do it virtually and give them this information and ask them to use paper and pencil to work it up, then it becomes very tedious and the students really don't like it. So if you're willing to sort of, it's very easy to just print out a string of multiple copies and then cut it at different places. And I bundle them into Easter eggs and give it to the students, and then they're supposed to puzzle it back together. And that actually works quite well.
But you would need longer strings than these.
Yeah. So about a page long. Because I've done the exercise so many times and spent hours cutting the piece of paper, I thought of, can I get a printer that just does a string? So that's actually a good exercise if you do physical